we are going to discuss the muscle contraction. The first stage is called relaxed stage. Here there will be a Z line. This line is called Z line. Then in Z line we can find another type of structure, that is actin. This is the actin filament. Then in actin filament we can find some active sites, and this active site is called, this is the active site, and this active site is filled with a structure. This is one more active site, and these active sites are filled with a structure called troponin. And here we are taking a myosin filament and one more actin. In myosin filament we can find a structure, that is myosin head or head of myosin. So in relaxed stage is the first stage of muscle contraction. Here there will be a Z line in an actin filament. In actin filament some active sites, here we are taking 3 active sites, and these active sites are covered by a structure called troponin. Then there will be a myosin filament. In myosin we can find a structure or projection called head of myosin. Then we are taking one more actin in opposite side. So we should note that there is no connection between actin and myosin here. That is, there is no formation of actin myosin cross bridge here. No actin myosin cross bridge. Actin myosin cross bridge means connection between actin and myosin. So here there is no connection between actin and myosin, so there is no actin myosin cross bridge here. Then we are going to consider the second stage. The second stage is called stimulated stage. Again in stimulated stage or during stimulated stage, the muscle fiber will receive stimulus, and due to this receiving of stimulus, the T tubules will release calcium ions. Here we are taking a Z line, an actin filament. In actin filament we can find some active sites like this, and these active sites are filled with troponin, and we are taking a myosin filament, and we are taking one more actin. So here we can find the troponin. Also the troponin will also present here, okay, and I am placing this released calcium 2 plus here, and this is the myosin, this is the actin, and these 3 are the active sites, and these 3 active sites are filled by a structure called troponin. We already discussed it in previous stage. So During this reaction, the released calcium 2 plus due to the receiving of stimulus, this calcium 2 plus or this, so the calcium 2 plus is released during receival of or receiving of stimulus, and this troponin has higher affinity towards this calcium 2 plus ions. So the troponin from this active sites will release from this active site will release from this active site, and it will combine with the calcium 2 plus. So the troponin from this active site is removed from this active sites, and the resultant structure will be like this. This is the Z line, and this is the active site, this is the myosin, and this is one more actin. So here we can find troponin, here also we can find troponin, but the troponin from this active site is removed and combined with the calcium 2 plus. So this active site is free, and during this time one molecule of ATP will hydrolyze, and which will convert into ADP, an inorganic phosphate. During this reaction, some amount of energy will release. From this energy, a small amount is using for the formation of actin myosin cross bridge. We already discussed in previous video, and the remaining part of energy will be used for the attachment of myosin head at the free active sites, that is, and this connection between actin and myosin is called actin myosin cross bridge. So the energy released during hydrolyzing of one molecule of ATP is using for the actin myosin cross bridge formation, like this. Also here here we can find the actin myosin cross bridge formation. So out of this energy, a small energy or small amount of energy is using for this actin myosin cross bridge formation, and the remaining energy will be used for another reaction during later stage or during coming stage. Okay, then we are going to consider the third stage. Here we are taking a Z line, this is the Z line, and we are taking one actin filament, this is the actin filament, and we are taking a myosin, this is actin, this is myosin, and one more actin here, and the myosin head, this is the myosin. On myosin we can find a myosin head, and this myosin head is bind with the active site of actin. During the previous stage, the myosin head will attach with active site of actin filament. So we know that one molecule of ATP will hydrolyze and which will convert into ATP and ADP and PI, and during this process some energy will release. So out of this energy, a small amount of energy, out of this whole part of energy, a small amount is using for this actin myosin cross bridge formation, and the remaining energy is free. And this free energy or remaining energy is using for the rotation of myosin head from one position to other. These are same myosin head, and by using this energy produced by ATP hydrolyze, the myosin head is moved from first position to second position, and this rotation is called power stroke. Very important. And this rotation is called power stroke, and which is given by head part of myosin. And this rotation or power stroke by using energy is provided by head part of myosin. By this power stroke, the actin filament will pull towards the center. Actin filament will pull towards the center. So the distance between two actin filaments were reduced. And we know that the actin filament is associated with the Z line. Actin is associated with the Z line. So this Z line will also move towards the myosin. The resultant Z line will be like this. So the distance between Z line and myosin filament is reduced. Also the distance between two actin filaments were reduced. So this reduced, okay, and by this power stroke the distance between two actin filaments reduced, and the distance between Z line and myosin filament were reduced. Then we know that the power stroke is given by the energy produced by ATP hydrolyze. The energy for this power stroke is given by ATP hydrolyzed, and by using this energy the rotation will be there or power stroke will be there, that is rotation of myosin head from one position to other. Here we are saying that from first position to second position. But after finishing this amount of energy, the myosin head will return back into its original position, that is from second position to first position, and this process is called